Hello, this is Miss Dagenford, and I wanted to get you started on dihybrid crosses. And in class, we left off with the example of using blood typing as well as sickle cell anemia to show the idea that you can think about two different genes, not just two different alleles, which that would, when we're talking about a tall plant versus a dwarf plant, those would be two different alleles of the same gene, plant height. Well, in this case, we're talking about two different genes, the gene for blood type and the gene for hemoglobin, whether or not it's normal or sickle cell. So you can imagine this starts getting a little bit more complicated because you're trying to determine all of the different possibilities. So let's start off easy. And so these dihybrid crosses, this is a very classic example, and this is what Gregor Mendel, he did quite a number of his experiments with pea plants. And so in pea plants, if you have a round pea, that's dominant over a wrinkled pea. You could also have the pea color, and that would be yellow, which is dominant over green. So those are our symbols, big R, big Y, little y. Now, if we just take a very basic cross, homozygous dominant for both traits and homozygous recessive for both traits. So this would be one parent and this would be the other parent. Now, remember, during meiosis, you reduce the number of chromosomes. Each of the daughter cells gets just one of each homologous chromosome. Now, in this example, it doesn't matter really how they line up. The daughter cells, once they separate in anaphase 1, each of the daughter cells would get either the big R and the big Y, or in the other parent, it would get the little r and a little y. Now, I'm skipping over a part that you might need to remind yourself. Remember, these are duplicated chromosomes. So not only do the homologous pairs separate, but then in meiosis 2, those chromosomes then split. And you can go back in your notes and review that if you need to. So in this case, you're going to set up a Punnett square using these gametes. Now, the offspring, this one's a little obvious. All of the offspring are going to get the big R and the big Y from this parent, and a little r and a little y from that parent. So all of the F1s would be big R, big Y, little y. And if you want to do the Punnett square to show you this, and because the gametes are all big R, big Y, and little r and little y, when you drop and cross, keep the letters together. So the R's stay together and then the Y's stay together. And that's how I did my very easy Punnett square for this one. Now, what if we were to cross two F1s together, and this situation gets a little bit more complicated? Now, in plants, it's pretty easy. We can uh, cause them to self-fertilize if they are able to. So we could take our F1s, which now are heterozygous, and we can cross them. Now, here's where you want to think about all of the possible combinations. Now, there's a couple of ways to think about this, and I want to make sure you understand why this happens. So let's take this case over here. This plant, remember, there's two sets of chromosomes that we're referring to here. At least we're that's what we're going to presume right now. This chromosome would have the big R and big R, and then over here would be the little r, little r. The reason why we have two is, remember, they duplicate when they go through meiosis. And then we have another set of chromosomes with the big Y and then the little y. Now, here's what you gotta remember. They don't have to line up like this. So let's think about scenario one. If these chromosomes lined up like this, when anaphase 1 happens, you would end up with two gametes, big R, big Y, and then little r and little y. However, what if they line up Let me clean that off a little bit. What if they lined up differently? What if, and I'm just going to do this really quickly here, what if they lined up so that the top two are the same? So big R, little r. But what if the bottom two are little y, big Y? If you did that, then notice as they separate, you're going to get two different types of gametes. So let's do that. Let's separate them out. If these then separate during anaphase 1, now you would get daughter cell, or I'm sorry, now you would get gametes, an egg or a sperm cell with big R, little r, big Y. Now, there's the way to look at it through meiosis. But you can also do this in a somewhat mathematical way, looking at all the possible combinations. A lot of people like to think of how they learned foiling in their algebra class, and it's similar. You could say, okay, well, I could take this big R with this big Y. I could take this big R with this little y. I could have a gamete that has this little r and big Y, or this little r and this little y. So either way you want to think about it, you end up with four possible gametes. And when you make your big 16 square Punnett squares, you would put your four possibilities up top. And each parent, because they're both heterozygous, they're going to be similar. So big R, big Y, big R, little r, big Y, little r, little y. And the other parent's going to be the same because they're both heterozygous. Not doing this very neatly, I do apologize. Now, in a 16 square Punnett, setting it up is the easy part. It then becomes quite a tedious matter, although not hard, to drop and cross each square. You just have 12 more squares to do than a 4 square Punnett. So you'd have big R, big R, big Y, big Y, big R, big R, big Y, little y, big R, big Y, big Y, big, little. And you keep filling them in. And as you fill them in, you can look at what kind of plant that you get. In this case, you'd have round and yellow, round and yellow, round and yellow, round and yellow. Now, you can go ahead, and I want to check this when we come back to class, fill this in, and just to kind of save us some time, this is what you would get. Now, this is just kind of rotated, you know, a few angles, but this is what you get. You should get 9 yellow round, 3 green and round, 3 yellow wrinkled, 1 green and wrinkled. Now, if we want to prove it to ourselves, go back, fill in the Punnett square, and then fill in this chart. What you're going to do is figure out how many of these 16 are yellow round. You're going to prove it to yourselves. How many are yellow, green? How many are um, yellow wrinkled? My bad, sorry. Um, green round, and how many of them are green wrinkled? And you can just do a little tally. You should get 9, 3, 3, 1. And they should add up 16. If they don't, you counted wrong. So that's going to be the end of this video. I figured I'd keep this one short. I am going to give you another one because I want to make sure you get. I don't want to rush that one. So this is the longer. It's the more visual. Some of you will like this because it is very nice and visual. There is nothing wrong with this method. It does take a little bit longer, and I do think it's nicer to understand the mathematics behind it. So in the next video, I'm going to show you how to use probability to figure these out. It's pretty clever. And once you did understand this, you could do a tri-hybrid cross. You could do even more than that. And you can almost guess a tri-hybrid cross demands a 64 square punnet. Nobody wants to do that. So um, this should help you see why we set up the Punnett squares the way we do. By all means, sit back, look at your Punnett square that you completed, and if you're not finding that 9 to 3 to 3 to 1 ratio, one of the common mistakes is your Y's are not clear enough. A lot of people forget to make the big Y big and the little y little, and then it's easy to misinterpret that. Um, so, see you in the next video.